Right. So, hi everyone. My name is Emiel Wambo-Becker, and I'm 16, grade 10. I was a TKS Toronto student this year, and I've had the opportunity to engage in consulting challenges with Instacart to install Internet of Things devices and systems in stores for more accurate and timely listings. And I've also worked on a recommendation for the United Nations to get more women and girls in the digital economy in Vietnam. And then I also developed a solution for the mortgage underwriting process with quantum machine learning for a little hackathon we had. And most recently, I was working on a moonshot company to accelerate clinical 3D bioprinting. And we essentially are pioneers in the predictive tissue engineering space by combining bioprinting and machine learning. So I've learned so much about respecting the team culture and contract and ensuring transparent and honest communication, and also how to seek a field perspective and get expert validation, as well as how to take better feedback and give better feedback and to take initiative and be a better leader. So right now I'm working on a couple social impact projects, notably one with a team who is also in the Knowledge Society who are working to prevent maternal mortality in rural Nigeria. And they're planning to do this through the distribution of life-saving medicine, which is called misoprostol, and the upskilling of midwives. And I also dabble in science communication. So I love to publish Medium articles and write YouTube video and film YouTube videos, as well as work as a freelance writer. And finally, with TKS, I am also part of the iGEM team, which is the International Genetically Engineered Machine. And it's just the world's largest synthetic biology competition for students in the pursuing undergraduate studies to community labs. And we're all tasked with developing real solutions for pressing issues. And right now I'm looking into microbial communities that are near plants to reduce the use of chemical fertilizers and pesticides. And really by the end of this presentation, I want to introduce to you synthetic biology, where biology, engineering, and computing collide, and encourage you to explore the field further. I would also like to point out how failure is often an opportunity for growth and how we're really failing our way to success in biology. So I'd love to get your responses in the chat, but what do we have in common with sunflowers, frogs, and apples? Feel free to drop your responses. We'll see if we can get this. Okay, great. Yeah, we do have cells. Our cells include DNA, so right on that, Isa, and thank you, Timmy. But we carry our genetic information in the form of DNA to grow, change, and reproduce. So deoxyribonucleic acid is essentially a long, complex molecule that holds the instructions for making all the proteins in our bodies like a recipe book. Reading DNA means determining the order of 4 chemical building blocks or bases that make up the DNA molecule. And so about 20 years ago, researchers from around the world sequenced and mapped the human genome. That's about 3.2 billion base pairs. And since the cost of reading a genome has decreased dramatically. Editing gene sequences means cutting, removing, replacing, or adding DNA strands. And you may all be familiar with the CRISPR-Cas9 system, which is a technique to edit genes that is easier and more accessible than previous methods. And finally, writing DNA involves creating DNA molecules by joining together bases. And so gene synthesis methods allow us to construct and assemble genes from bases. And now with the ability to read, edit, and write DNA, we can harness the power of nature at the most fundamental level to solve the world's biggest problems in medicine, manufacturing, and agriculture. Thanks to synthetic biology. It combines advances in biology, engineering, computing, but also chemistry, molecular biology to make useful living systems. It's all about designing biological parts, devices, and systems that are already found in nature, but that don't exist at all. So it's going beyond what we know to redefine a new category of living organisms. It enables us to go from idea to product faster, cheaper, and with greater precision than ever before, because it makes biology easier to engineer. So just like electrical circuits can be made of various switches, transistors, and power sources, synthetic biological circuits are built from a range of genetic parts, and these allow us to or they're known as biobricks. And essentially, they are similar to Lego pieces because they enable synthetic biologists to take a workable and programmable approach and increase the speed of making biological products. So genetic parts consist of genetic codes, and think of it as DNA sequences. So you may be familiar with a bucket of Lego pieces, and essentially you have sequences online in a digital registry, and you can then synthesize them into DNA and then design, mix, match, and stack these Legos essentially for larger synthetic biological circuits, which can be incorporated in cell-free systems as well as host organisms. So synthetic biologists identify and catalog standardized parts that can quickly be used to build novel biological systems with improved predictability and robustness. And so thanks to the availability of tools to read, edit, and write DNA and the standardization of parts thanks to biobricks, synthetic biology is poised to significantly impact your life. And I'll tell you how. First is through bioremediation. Humans are notoriously messy, and I think we've seen this through oil spills or contaminated groundwater that we tend to harm our planet, and this becomes a problem not only for wildlife, but also for human health. And so bioremediation is the use of living organisms to remove contaminants, pollutants, and toxins from soil, water, and other environments. So naturally, bacteria and living organisms already decontaminate soil or water. They're capable of doing remarkable things, and others have been used for decades to detect and identify environmental pollutants. But let's say there are new types of contaminants we want to degrade or other species we want to develop. We can look at microbial biosensors to bring them further to target toxins such as, again, arsenic, cadmium, or mercury, or heavy metals, and respond in a variety of ways. So they can, for example, generate a signal or degrade a pollutant. And with synthetic biology, we could really harness microorganisms for bioremediation to clean our water, soil, and air. Next is manufacturing. So to manufacture existing and novel products in a sustainable way, synthetic biologists identify gene sequences that give organisms certain traits, synthesize them in a lab, and then insert them in other microorganisms like E. coli, so that they produce the desired proteins or characteristics. I actually wrote an article about just the development a company was making in the field of musk scents, and my article was published in Forbes, and I was really proud of it, but it was essentially how musk had been used or developed in a lab in a more environmentally friendly way. And it was just outlining the potential really synthetic biology really has to allow us to create new products, again, more sustainable manner, rather than using petroleum-based products that produce large carbon emissions and are again, environmentally detrimental. So biomanufacturing allows us to produce scalable and sustainable products ranging from medicine and flavor and fragrance ingredients, just like musk, and thanks to Isa for sharing the link, to bioplastics and biofuels. So soon, all our products may be bio-based for more alternative and more eco-friendly consumption. Next, I want to just present sustainable agriculture. So as I was mentioning, I am working with TKShowcase on how we can reduce the use of chemical fertilizers as well as pesticides. And we're looking to increase then crop yield as a result of increased nitrogen fixation occurring between plants and then bacteria. Climate change impacts such as higher temperatures, extreme weather, drought, and increasing levels of carbon dioxide, as well as sea level rises, really jeopardize the quantity and quality of our food supplies. If we don't do something now, who knows what our food systems will be threatened with later on. And synthetic biology may hold a key to the sustainability by providing, for example, the necessary tools to reduce agriculture reliances on chemicals. Again, as I mentioned, is what we're trying to do with TKShowcase, but also to enhance soil quality and improve global nutrition, while also providing an economic benefit for farmers. So all around, the potential for synthetic biology to revolutionize the space is really huge. And I finally want to touch on therapeutics and diagnostics. So synthetic biology also has biomedical applications, either through rational drug design, immunotherapy for cancers, or the creation of medical treatments with more sustainable practices. So for therapeutics and diagnostics, synthetic biological circuits can be designed to perform like a logic function using standardized biological parts, again, these biobricks. Biological devices can, for example, trigger a therapeutic or diagnostic response as output based on input signals they detect. So we could have little microbes in our bodies swimming around, identifying biomarkers and really tackling them before we're even made aware. They'll be really 10xing the immune system. So as genetic and molecular parts are discovered and characterized, synthetic biology will really usher the next generation of therapeutics and diagnostics. It's really about imagining the most outlandish and most ambitious goals, then finding a way to fulfill them with biology. Ask how to improve our transportation or manufacturing processes. How can we improve our living conditions and quality of life? These are all categories that are to be redefined thanks to this technology. So imagine all that's possible, if we leveraged what we know about biology and really just took our principles of engineering and computing and then to solve the world's biggest problems. My question for you is what would you build with biology? Whether it's specialized cells, again, that travel along your body to diagnose or treat a disease, or yeast that make biofuels instead of bread, plants that grow into tree houses large enough for people to live in, or algae sponges that purify contaminated drinking water. In the end, the best case scenario is that we engineer novel systems and that work reliably and address important world problems. Worst case scenario is that the systems we build fail the first time, the second and the third, maybe fourth and fifth, and they'll fail in possibly surprising and sometimes dangerous ways. But at this early stage in synthetic biology, both the successes and the failures have so much to teach us about how biology works at a fundamental level. Successes and failures advance our understanding of living systems and drive synthetic biology forward, and as a result, help us foster sustainable development. So let's fail, 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 and fail again to uncover secrets about life and living systems. Success usually comes as a result of past failures, so let's embrace them. Let's be fearless, and because every discovery takes us forward, let's fail fast and learn faster. I'd say that the biggest challenge in synthetic biology and what may be preventing it from really revolutionizing all these applications and industries I mentioned is our relatively limited understanding of biology, which leads to problems with predictable and robust design. Yet, if we continue to design, build, test, and fail, and learn, we have the opportunity to pave the way for success. Because when we embrace failure in synthetic biology, we can extend our knowledge of how living things function while pursuing our imagination and ambition. So if you have any questions, I'd love to take them now, and any comments are also welcome. Thank you.